necessary, and this is the structure of it. So later on when you guys look at action potential, or when we've already looked at it, but you just forgot because you let ethanol cross the semi-permeable barrier in your brain and killed your brain cells. You're going to see a lot of these things here, okay? A voltage-gated channel. So here we have the pore where your ions would actually be passing through, okay? Cell membrane, what would be important? The big difference, or the big thing you want to pay attention to in a voltage-gated channel is this inactivation peptide. So one thing that you'll find is really confusing, and you'll see it later on, is a voltage-gated channel has three conformations. It can be open, it can be closed, or it can be inactivated. Has anyone seen this yet? I guess some of you have seen it. So this domain here, we see two bulges here. These can, okay, let's say the default is closed. Okay, so these two domains kind of squish each other together. That's called the closed conformation. Alright? There's a change in the electrochemical gradient across this membrane. It opens. Now what's going to happen when it opens? Let's say this is a sodium channel. This thing opens. The concentration of sodium outside of the cell is higher. Where's the sodium going to go? It's going to move inside. So the concentration of sodium is going to increase on this side. What is that going to do to the electrochemical gradient across this membrane? It's going to raise it. It's going to become more positive or less negative. Okay? This is going to change the voltage again, a second time. Now what happens? This thing doesn't just close again. One of these inactivation peptides actually shoves itself right up there. Shoves itself in there and plugs it up. So this thing is inactive. Guys, you get your minds better places. Alright, so those are the three conformations, okay? Open, closed, or inactivate. Those are the main differences. So again, it looks like you guys look a lot at the structures. Here's a tetramer for a voltage-gated channel. They're all identical. Now again, this is going to represent a string of amino acids. They say that this string of residues here, 4, is what's responsible for actually sensing the potential difference across the membrane. Okay, this is going to be the little inactivation probe. Okay, this is what's actually going to move into the pore to block it up. And 5 and 6 are the residues that are actually going to be around the pore. And they're actually going to be responsible for opening and closing the pore in response to changes in electrochemical potential. Okay, here's a sodium channel. More or less similar. Here they have the inactivation segment here. The difference is where in the potassium channel, all the tetramers are identical. In the sodium channel, I think there are slight differences in the helixes. So we talk about monomer 1, monomer 2, monomer 3, and monomer 4. All right? I think that's the only little detail you might want to pay attention to. I don't even think the differences are that noticeable. It might just be a difference in the sequences. Yeah, here we're just showing it again. We understand how it works. You might want to look at the ball and chain diagram. So here are the actual parts that are going to be opening and closing. If you look at the S6 helixes, these are going to close. You can look in your book for more specific details about that. And what else do we have? This is a top view showing potassium flowing through the pore. I won't go into too many details here. You can look in your book, and you know better than me what Dr. Zerges is probably going to expect from you on the exam. I don't know if he's going to ask you for specific residues, but you can talk to him about that. Again, I think maybe you can look in the book here. What they're specifying is, again, these are going to be amino acid residues, and these residues, which are going to be positively charged, I'm sorry I didn't write it down. I can't remember which amino acid residues are responsible for detecting the change in voltage across the membrane, but you'd expect them to be highly charged. Apparently they're positively charged. So somehow the change of, excuse me, the change in electrochemical potential across the membrane is going to be detected by this, and somehow it's going to change the conformation of the proteins in the helix. Is everyone okay with that? Again, this is more molecular stuff. I'm not sure how much detail, but again, you can look at the P sigma, which we said was responsible for the selectivity of the region, as opposed to these S4s, S6 parts, and that are going to be responsible for opening and closing the channel. And there are 3 different models, so there may be a paper for you guys to look at online. Is that possible? A paper that you might want you to read that explains these 3 models? So anyway, I didn't see it online. Maybe he's going to put it up when he comes back. I won't go into too much detail about this either. Again, it's another voltage-based channel. What you guys, what they seem to place a lot of details on is the paddles. So I think that's what the paper is going to focus on a lot, is when they were talking about these voltage-gated panels. This thing looks like some kind of creepy alien. The paddles are actually moving the different regions of this channel into place in response to different voltages and things of that nature. And again, I think the most important thing that you guys are going to want to remember about these voltage-gated channels are the 3 conformations: rest, open, and inactivated. Okay, this is going to come up at least one more time for the course. All right? He mentioned this quickly when I was talking to him. This is the guy who discovered the ion transporting enzyme Na+, K+, DPase. He also won a Nobel Prize. And again, I think all of these things are going to be explored in more detail when you guys look at action potentials. Okay, and I explained what the action potentials are briefly, but I think that's a whole lecture in itself. I was going to sort of tack it on to the end of this, but there's only 15 minutes left. So, if there are any questions, you can ask. If you want to, yes?
Okay, I was just wondering, and maybe I missed out on it, but why do we need an inactive form?
The inactive form is because there's going to be a period of time where the electrochemical difference is going to have to balance itself out.
So
Right?
it still needs to be open for that period of time.
No, it's going to be inactivated. Nothing is going to pass through. But there's going to be a change in electrochemical potential. So in other words, you don't want it responding to this gradual change back to its original state. You just want it to be blocked up. Does that make sense?
But why not close it?
Why not? In other words, you need 2 detectors. You need 2 responses to a change in electrochemical potential. Now you can respond firstly to the rise in positive ions on one side. That's going to close it. The next thing you need to respond to is the rise. So do you see what I mean? So if the potential is lowered to such an extent that it just closes it up, when it starts rising again, it will open. So you need to block it while it rises back to its resting potential. Does that make sense?